Okay, everybody, welcome to the review for the chapter 4. This is about the prokaryotes. And again, remember to go back and actually watch the longer lecture for all the information that you might need to know for the exam, because this won't cover everything. So prokaryotes are organisms whose cells do not have membrane-bound structures. They'll have a lipid bilayer surrounding them as part of their inner membrane. All have that. But there's no other membrane-bound things in their cells, okay? That's prokaryotes. Pro meaning before, karyote being nucleus. So before nucleuses came around, okay? All right, so they all have something that we would call the cell envelope. Some of them have so everybody has an inner membrane. So that's a lipid bilayer. It's just pretty typical lipid bilayer, like we had talked about in the chemistry chapter. And then there is a cell wall. I'm not going to go all the way around, guys, please. And then some of them also have an outer membrane. Okay. We'll just say that this is what we're going to be looking at. Okay. So that's that whole thing is the cell envelope. Okay. Cell wall of bacteria, prokaryotes, cell wall of bacteria are going to be made up of peptidoglycan. Okay. Peptidoglycan, peptide, that's like amino acids linked together, right? So a peptide or peptide chain is literally just amino acids linked together. This is a small chain of amino acids that are linked together to hold together a repeating units of these types of sugars. NAM and NAG are certain kinds of sugars. N-acetyluramic acid and N-acetylglucosamine. So they're basically sugar-based structures. That's the glycan part. And then peptido is the cross-linking of the peptides that hold it all together, like a scaffolding. Okay, so NAM, NAG, all linked together, whatever, they're bound together. And then these little peptide chains to kind of link it across to the next one. And then you could see how that could build up over and over and over again, essentially like a scaffolding. Okay, that's the peptidoglycan cell wall. Gram-positives have a very thick cell wall, multiple layers of this going over and over and over again. And it is in fact stabilized and made stronger by lipoteichoic and teichoic acid. So that helps them whenever they are dividing to keep everything, when they're replicating everything, to keep everything steady and stable and all that. Okay, so the gram-negatives, they have a very, very thin layer, like a single layer of peptidoglycan. It's super thin, and they therefore need a little bit extra support. So they have another layer. They're the ones that have this extra outer membrane. So that's the gram-negatives, whereas the gram-positives have this thick cell wall. Okay. All right. Acid-fast bacteria are gram-positive. So these were our Mycobacterium and Nocardia species. Mycobacterium is the one I want you to be more concerned with. Mycolic acid, that waxy substance in that gram-positive wall, makes them very hardy. They can survive harsh conditions because of it. They do not have endospores, but they may as well be endospores. Okay. Next is the mycoplasma. These are another special group. So we have the gram-positive, thick cell wall, gram-negative, thin cell wall, extra outer membrane. Acid-fast, mycolic acid in the thick cell wall. And then mycoplasma, no wall at all. They are pleomorphic because they can't maintain their shape. Pleomorphic means that they can have several different shapes to their cells. Their cells can be all sorts of different shapes. All right, speaking of cell shapes, the basic ones that you guys need to be aware of. The cocci are the round ones. The bacilli are the rods. So cocci are like little spheres. The bacilli are rods. And then we have different arrangements like staphyla, which are the grape-like bunches. Okay, and then strepto, which are chains. We have diplo, which is just like in pairs. like Neisseria gonorrhoeae is Diplococcus, so they exist in pairs, like little, just like that. There's other ones like bacilli also have these ones like called palisades, which is like they, are attached like fences. A palisade is a fence. So that's what they look like as fences. So there are other ones as well, but these are the main ones that you know. Movement and flagellar arrangements. Okay, if a bacteria can move, it uses flagella. That's the only thing that allows it to move. Fimbriae, attachment. Sex pili, attachment and or transfer of genes or something. But movement, that is only flagella, okay? So we have polar, which could be at each end. We have amphitrichous. We have monotrichous. So amphitrichous would be like at each end of tufts at each end. Little tufts at each end. Monotrichous, you have one flagellum, and that's it. Peritrichous, covered all over with flagella. So those are some examples of terms associated with flagellar arrangement. Flagella look like little whips attached to their ends, and they help them move around. They move via rotation, not by whip movement. They look like a whip, they move by rotation. Capsules. Capsules are protective for prokaryotes, for bacteria. Capsules and slime layers, basically they're part of something called a glycocalyx. Write it down. So it's a sugar-based structure on the outside of a cell. It could be sugar and peptide-based structure on the outside of a cell that could be loosely associated, which is what we see in a slime layer, or tightly associated, so it's more stuck right on the outside of it, and that's a capsule. Capsules are for attachment as well as to evade the immune system for something called phagocytosis, which is where our cells try to eat up the bad guys. Okay, phago being eat, cytosis eating cells, cyto cells. Okay, so yeah, that's capsules. So some organisms have that. This usually the ability to form a capsule or some sort of glycocalyx type of layer, whether it's a slime layer, that can allow them to stick to things as well. So they can stick to things, then other things can come and stick to them, and then can all grow together and form a film. If you have ever had plaque or ever needed to ever brush your teeth in your life, then you know what a biofilm is. This is just where bacteria kind of grow and stick up on top of one another and create a little commune like that. They can send little signals out to one another to let each other know how the commune is doing. So that is called quorum sensing. So holding a quorum, people that want to talk about something, that's what bacteria do. It's quorum sensing. Okay. Endotoxin, this is part of that outer membrane. So gram negatives, remember, they have the outer membrane. Gram positives do not have this. The gram negatives have the outer membrane, and it has something in that outer membrane called lipopolysaccharide. That is referring to endotoxin, which is something in the outside of a gram negative membrane. Our cells, our body recognizes endotoxin as bad guy stuff. Okay, it's part of what we call a PAMP, which is a pathogen associated molecular pattern, and our cells immediately recognize that as foreign. It should not be there. It also is fever inducing and all this stuff. Endotoxin is going to make a very, very common reappearance throughout this class. So be aware of endotoxin and what it is, and that it is associated with gram negative bacteria. All right, plasmids. These are little bits of circular DNA that are much, much smaller than the genome. They don't code for any essential genes, but they can code for things that can help the bacteria survive better. So things like antibiotic resistance can be coded for by plasmids, and they can be transferred from one cell to another. All right, next is the endospore. We had mentioned this in the previous chapter. This is something that bacteria, some types of bacteria, Bacillus and Clostridium. So Bacillus, example of a pathogen would be Bacillus anthracis that causes anthrax. For Clostridium, an example of a pathogen would be Clostridium difficile, C. diff. So both of these are endospore forming pathogens. That the fact that they form endospores is like a characteristic of that disease, right? So it's very important to be aware of that because it's very hard to get rid of endospores. They're extremely hardy. So vegetative cells are the normal metabolically active cells, just typical dividing normal, active cells. Sporulation refers to the process for making the spores. And when a vegetative spore shifts into sporulation to start making endospores, it becomes the sporangium. So that's the cell that's surrounding the forming endospore. Okay. But yeah, so then the endospore is made, and then, whatever, it goes off and relaxes in the environment until more food is around or whatever, and then it will germinate. Germination is the process through which that endospore becomes a vegetative cell again. And it basically just sheds its little outer cortex, its covering, and just becomes a vegetative cell again. That's endospores. Okay, so that is that for the prokaryotes. We'll come back for the eukaryotes for the next video.